All right, today we're going to begin our kind of ecology unit, and we're going to actually start talking about plants, because plants are kind of the sometimes unsung heroes of ecology. And actually, this is one of my favorite units because, you may never take a science class again after this, some people. Um, but most of us in our lives are going to be enjoying the out of doors and natural areas and going to parks with our families, whether the family you have now or in the future when you maybe have your own family. Um, and so understanding about how the environment works and how you impact the environment and how it changes and it's all related is something that even, it's going to be part of your life kind of forever. Whereas, maybe our talk about cellular respiration won't be. So I think it's one of the most useful units. We're going to get started today about talking about plant responses, and tomorrow we'll talk about those in animals. And uh, one of the things that's hard for us to realize is that plants need to respond to changes in their external environment just like animals. So animals might do this by moving away or towards different stimuli. And while plants generally can't move quite the same way that animals do, they need to adjust their growth and their development in a similar way. So we have two pictures of ways that plants might uh, respond to different stimuli. We'll look first kind of at this picture right here with the potatoes. So obviously, these two potatoes are responding to a stimuli. The one on the left was grown in the dark, and the one on the right was exposed for 1 week to the light. And there's differences, I think, pretty obviously in their growth and development, just based on the light. So light as a stimulus has caused both of these potatoes to react in a different way based on if the light was present or if the light was absent. Now, at the smallest level, all of these responses work based on signal transduction pathways that we talked about during the cells unit. So just remember, kind of on a basic level, the idea of a signal transduction pathway like we talked about then. Again, this is a good place to insert some review. And so all the responses we're talking about are going to be in this response phase of signal transduction, and there will be transduction elements in a reception phase as well. So as an example, with the potatoes, okay, light is the signaling molecule, and there's a phytochrome receptor, which is the part of the reception phase. A secondary messenger, whether it is C-GMP cyclic guanine monophosphate or calcium ions, are the transduction molecules. They're the messengers. And through a protein kinase cascade, like a phosphorylation cascade like we studied, they activate transcription factors which cause greening, what's called de-ethylation. Okay, so all of our examples about plant responses we are going to talk about are linked to the signal transduction pathway. Not this one specifically, but a signal transduction pathway. So we won't look at each of them this closely, but we should keep in mind that they all operate similarly like this. So this is one example. That's how we get these differences. At the heart of most plant responses are what we're called plant hormones. So just like animals, like humans have hormones, which are chemicals produced in kind of small amounts in certain areas of the body and that travel through the blood or through the vasculature to activate organs on different ends of the body to trigger responses. Plants have similar chemical compounds. Okay, so hormones exist in humans and animals and in plants. These are kind of the uh, big plant hormones. And so if you're interested in plant physiology, you'll learn a whole lot more about these. We'll talk about auxins, gibberellins, abscisic acid, and ethylene here today as examples of plant hormones and ways in which plants respond to stimuli. One of the most obvious things that plants do to respond to changes in their environment is called what's phototropism, where they grow towards the light. If you've ever had a plant that you put like on a windowsill or next to a window in your house, you continually have to rotate it to keep it kind of growing straight so it doesn't just skew one direction and look kind of interesting. And so here is an example of an experiment done about phototropism in 1880, trying to discover what causes phototropism. And so looking at this experiment, one conclusion that was made was that, there must be some sort of chemical uh, intermediary to control phototropism, and it must originate in the tip, kind of like you look at on the right side of the second experiment in 1926. The tip is kind of the key. What these experiments eventually discovered was auxin. Auxin is just another name for indolacetic acid, or IAA, which stimulates cell elongation, and it's produced in what's called the shoot apical meristem, or that's the area of rapid growth in the shoot, the tip of the plant, and that's kind of like a growth plate in humans. Similar, just trying to find some sort of analogy. What happens is that the auxin is not distributed symmetrically. It accumulates in cells on the darker side of the plant, and it stimulates elongation of those cells. So if those cells are elongating and the ones in the light aren't, it will naturally cause the plant to bend towards the light, and then we get phototropism. So that is one key intermediary to the phototropic response, an example of how a plant is responding to the stimulus of light. Here's a picture of a plant, and here's the apical meristem, and then we have what's called a root ground meristem right here in the roots. They're both areas of rapid growth and development. And this one in the shoot is where we talked about auxin being produced. Gibbsellins are a large group of over 100 different molecules that have many effects, like promoting stem elongation, fruit ripening, or seed germination. And here in this picture, you can see how when a seed is in the ground, we wouldn't want it to grow if there's no water for the roots to tap into to make a healthy plant. So when water is able to permeate through the seed coat, it activates gibberellin within the seed and promotes the gibberellin, that is, becomes distributed and promotes alpha amylase production, which breaks down the endosperm, the nutritious part of the seed, into sugar, which the plant can use to grow, like you see on the far right. So gibberellin can make sure that a seed won't start to germinate until it's able to grow. Abscisic acid is another example which affects the slowing of growth, and it promotes seed dormancy. And again, it makes sure that a plant seed will survive, and that will only germinate under certain conditions. All right, only under certain conditions. Ethylene may be one of the most obvious plant hormones, is multiple effects, and it's really important in the response to mechanical stress. And it's in charge of what's called the triple response for mechanical stress, which is to slow the stem from elongation and become thicker, and to start to curve horizontally to make it harder for herbivores to eat the plant. It's also involved in apoptosis, so programmed cell death, getting rid of old and unuseful cells, and leaf abscission, so leaves falling off during the fall. But maybe most obviously or testable to us, it's used in fruit ripening. So fruit are really important to plants because hard, tart fruit protects developing seeds from herbivores. So that's why while they're developing, fruit is really hard and tough and doesn't taste very good because the seeds are still developing. But when the seeds are developed and ready to be spread, fruit ripens to become sweet and soft, so it's attractive to animals because animals need to eat the fruit to disperse the seeds. Ethylene triggers fruit ripening, breaking down cell walls and softening it and converting starches to simpler sugars in the fruit. All right. Um, One of the things that this is useful for to us is that there's truth in the old wisdom one bad apple spoils the bunch because the ripening of a single piece of fruit will produce ethylene that will instigate ripening of other fruit. That's why it's one way to really quickly ripen green bananas is by putting them in a bag with a ripe apple. That ripe apple is producing a lot of ethylene and will ripen the green bananas. It's also useful in transportation of commercially sold fruit because commercially sold fruit is usually stored in high CO2 environments to kind of suppress ethylene production to keep the fruit ripe longer. Besides phototropism, there are other important reactions that plants have to light, such as photomorphogenesis, so how light changes the plant growth. Um, and these responses are really dependent on the plant being able to detect light, its intensity, its direction, and its color or its wavelength. Okay, and so one of the really important detectors of these different parameters of light is called phytochromes. Phytochrome is a receptor in cells and plants, and it's like a molecular switch. So it can have two states, kind of the PR state and the FR state. And the PR state stands for red, and FR stands for far red. So red light is light that has a high concentration of like red wavelengths. Okay, this is usually what you'd find in an open sunlight. And far red light has a high concentration of farther red, deeper wavelengths, what you would find in shade or in darker regions. So, for example, if the phytochrome is in the PR state and sunlight shines on it, it will have a high concentration of red light, and that will stimulate this phytochrome to switch to the PFR state, which allows for seed germination, flowering, and branching, and because there's a lot of light present. Now, if the phytochrome is in the PFR state and it is starting to receive a lot of far red light, it will switch back to the PR state, which will inhibit responses and stimulate other responses like growth and height, uh, so that it can get out of the shade and into open light. So examples of the phytochrome helping mediate these different light responses. One thing that happens is because it is the flowering response. So triggered by like photoperiod, it's called photoperiodism. So different plants might be called short day or long day plants based on the relative length of day or night. All right. In both of these cases, the night length becomes what's called a critical period, which is a trigger. So in short day plants, uh, like you see here, they will not, um, what should we call it? They will not bloom until the length of darkness, uninterrupted darkness, is beyond a certain critical length. So here, far left, it's not below that period, doesn't flower. In the middle, flowers because it's below that critical period. But we can manipulate that by artificially inducing flashes of light in the middle of that darkness period, and then neither of these segments are quite long enough to instigate it to flower. On the other hand, on the other hand, plants called long day plants will only flower if the length of uninterrupted darkness is below a certain threshold. This threshold. So here, Long day plants flower. Here, they don't because the length of nighttime is not below that threshold. And we can likewise stimulate these ones to flower by adding a flash of light during that dark period so that there is no uninterrupted dark period that is uh, that is in that period. Okay, uh, there there's there is no uninterrupted period of dark that's longer than the threshold period, so it flowers. This is a key way that in the industry of producing flowers, commercial flower industry, we can create blooming flowers in the middle of winter that normally in the wild would never bloom in the winter. Plants also follow some circadian rhythms, so just like humans have a biological clock, so do plants, and that they can detect different times of day, 24-hour cycles, and so they can maybe flower at different times of the day, or turn different colors at different times of the day, or have different leaf morphologies at different times of the day, and those are examples of plants following circadian rhythms. Plants also respond to gravity so that shoots and roots grow in the correct orientation. So roots have what was called positive gravitropism. They grow towards gravity. And shoots have negative gravitropism when they grow away from gravity. And these responses are really mediated by these starch grains called statoliths that you see on the right. And their settling in the cell one way or the other in accordance to gravity helps to instigate these gravitropic response and distribute growth hormones in that way. Some plants respond to touch, which is called thigmotropism. For example, if you've ever seen the mimosa plant, it's very sensitive to touch and its leaves will scrunch up when touched. And that's caused by a change or rapid loss of water and potassium. And then lastly, plants have a number of defenses against herbivores and other predators, like thorns or toxic chemicals or unpleasant substances that they secrete to defend themselves. So here is an example of a parasitoid wasp larvae that's emerging from a caterpillar. So in this case, the plant has a symbiotic relationship with these wasp larvae that help defend against herbivores, these caterpillars. So in this situation, um, when the sharp leaves of the plant wound the caterpillar, a signal transduction pathway within the leaf signals the synthesis of a volatile attractant, a chemical that will attract these parasitoid wasps that end up laying their eggs within the caterpillars. I know this is really maybe unpleasant to think about, but it's one example of plants defending themselves against attackers. There are also a host of responses that plants have against pathogens. So there's like, for example, a plant immune system almost. Um, and this is a lot for us to get into. I wanted to mention that it is present. You won't specifically have questions about certain mechanisms at all in plant immune systems and defenses, but it is important for me to bring up that they exist. Right, and you can kind of look at this slide and we can discuss more in class. I didn't want to just spend a whole lot of time with it since it can get very complex, and for the exam we won't need to know a whole lot about it.